Hi everyone, I'm Santiago Ocampo.
And I'm Maria Monica Noguera.
And today we will be presenting you this topic.
First, let's talk about the Staphylococcus pseudintermedius. Although the Staphylococcus pseudintermedius are common bacteria of dogs, they are also opportunistic pathogens, being the primary cause of dog skin and ear infection. Recently, more attention has been given to this microorganism because have been showing and the science have been discovering resistance to methicillin and some antimicrobial treatments. It's important to humans and for the medicine because it could be a zoonotic infection, but the science is trying to discover what the Staphylococcus pseudintermedius in specific could create. Because what we know is what the Staphylococcus family could create.
One of the great problems today is a great emergency of the resistance to typical antibiotics. Bacteria may have a natural resistance to some family of antibiotics. This resistance depends to the variability genetic that suffers in the evolution. Also, the bacteria can be acquired. This resistance can be different mechanisms such a small or large mutation in this DNA and different mechanics for subsistence and protection against to the antibiotics, such as generate an enzyme that destroy the chemical structure.
Okay, the goal with this study is to search and analyze, based on previously known dynamic of virulence, how this microorganism, the Staphylococcus pseudintermedius, can be resistant to antibiotics and to, in specific, the methicillin.
PCR is a technique of lab for amplify sequence of DNA. In this case, it was used for sequence and show the genes that are related with the resistance of the mechanisms.
Okay, the multi-locus sequence typing is a technique based on the sequence of different genes analyzing variation in different locus, directly analyzed by the sequence of the genes. And this could give us information about the relation or not in different microorganisms. In this case, it was used for the characterization of the pseudo-intermedius, and the thing is that also they were analyzing the enzymes and the components and how in their absence or in their presence could be or could create pathogenicity.
CDS page is a technique for separating proteins according to the electrophoretic mobility. In this case, it was used for analyze the purified proteins for check the theory that the pseudo-intermediates could alter the DNA from nuclease, for finally convert in the DO cytotoxin.
Cellular chromatography is a technique that consists in the separation and the qualitative analysis of different components and see the interaction capacity and their behavior in other substances. In this case, was used for analyze the migratory position of the DADO and the DAPP.
In this red circle, we can see the bad activity of the mu B. Here we can see the compare of the activity, the wild type than the mu A and mu C. They have similarity, but this important mu B. They have a darkness band, and for example, the band spreads different than the cow than the human. This is important because during the experiment, search remove this gene. And this we can see when remove the mu A and mu C don't show important result. But when remove mu B, they have a decrease of activity the nuclease. Here we can see the movement of the recombinant will be, because in this study we are looking for the enzymatic activity.
Okay. Here we are analyzing how the recombinant nuc B in presence of these components is their behavior and how can be their activity analyzing the chromatography. For example, the calcium chloride, if we see have presence, and for example with these agents, if we see doesn't have presence or activity in the chromatography. Different here that, for example, if we add this component, also are not present. Here we got like the degradation of DNA with the recombinant nuc B. If we see the cow and the human are the same chromatography, but change in the canine, because the microorganism analyzed has this component that degrade DNA, and of course, as I told you before, these microorganisms have been studied in canines, and of course can create infection mainly in dogs.
Here again we can see the movement by the recombinant DSA. In this study is one enzyme that have been studied for this relation with the cellular intermediate because it can create cytotoxin. Here they evaluate the relation with the phosphate inorganic during the enzymatic reaction.
Okay, in this graphic what we got is the hydrolysis of DNPP to the DO in having these components in the reaction. So if we see here, we're trying to analyze how each component and the activity in the chromatography. Here we see that if we got the three components from DNPP, we could have a hydrolysis and have as final product DIDO. That is important because DSDIDO is one important enzyme of our microorganism that can create cytotoxicity. Here what we got is like the enzyme activity, right? So we got here a mutant ADSD8, and this tells us that if we reduce this activity, there aren't like enzymatic reaction. So this means that our microorganism in fact need this activity to create cytotoxicity. Finally, what we want to do is to compare the research that previously made these authors about the topic with the article and see if the opinion is related or not.
And what is related with the article? They agree with the investigation.
And that's it. In conclusion, the molecular biology methods could give us a good help in order to, for example, have diagnostic methods. Because the article shows us so many different methods in which we can compare the, for example, cytotoxicity of different species. And comparing different species, we could create and analyze a good diagnostic for example, the Staphylococcus pseudintermedius in this case. For example, we got the pathogenicity, and with the molecular biology method, we can analyze the pathogenicity of, for example, one microorganism. Knowing that, we could create a specific treatment because we know the specific enzyme or the specific component of that microorganism that is affecting the biological walls or the immune system. And finally, the zoonoses are pretty important because in this case we could see that in this case the Staphylococcus intermedius came from a dog. So in this way we could analyze how in the dog have the behavior, and we could infer and try to guess how could be the behavior in the humans.